Arvind Van, and he's a computational biologist, and he's at Argonne National Laboratory, and he leads efforts to automate biology research laboratories. So I guess you'll be putting a lot of us out of work, huh? Thank you.
Oh, that's one way to think about it, but all right. Thank you. So, yes, I will talk a little bit about how we have been thinking of doing biology in the lab. I'm not a wet lab biologist. I started out as a computer scientist, got trained and interested in biology, started doing biology, and this is where it led me to. So I'll sort of highlight what we are going through, and this wouldn't be possible without all the collaborators and people that have been like working hardware support by Cerebrus. Of course, Anima is here. Um, and then a lot of people from Argonne and U Chicago, my grad students and everyone that have been involved in it. And this is actually their work more than my work. Again, from the visualization team, the data storage team, which actually provides us with the infrastructure, and then finally the robotics engineers to whom a lot of this work is actually like possible. So my group actually works with a class of proteins that are particularly hard to deal with, which are disordered proteins. And disordered proteins for the longest times were not even acknowledged as part of being important or whatnot, because there were no tools to actually study them. So the unfortunate problem, though, is that over 30% of the human genome itself is disordered. We know that they are disordered, um, and we know that they actually don't form a stable structure when it comes to like binding to their partners. They're highly flexible. They adapt their binding surfaces to various targets. And over 65% of these proteins are implicated in some form of function in diseases. So cancer, neurodegenerative diseases, cardiovascular diseases, diabetes, you take them, they're actually quite important. What is interesting to note is that if you were to take a look at a particular protein like P53, it has over 300 interactions that's known in the human proteome. This is validated through experiments. And in many cases, it can actually adapt its structure as an alpha helix or a beta strand or just as a loop, and it mediates the context of this signaling pathway that is actually involved in. So my group largely focuses on how we can actually understand how we can target these undruggable parts of the genome. And this is not primarily restricted to human genomes in any matter. In fact, viral, bacterial, and fungal pathogens actually use these disordered regions to mediate very big portions of their signaling pathway. Right. So this is actually the group's focus. And if you look at this from 2015 or so, in an ongoing collaboration with Richard Krivaki's group at St. Jude's, we were looking at this particular protein called P27, and in fact, a very particular, very small domain of about 25, 30 residues or so that was involved in being disordered. And they were finding like small molecules could modulate this really well. And if you look at why we needed to study them, we used simulations, massive amounts of simulations. And I'm not going to go into the detail, but you can read it in those papers, where we actually simulated many of these class of molecules for over hundreds of microseconds long. And we had to study them in the context of understanding how they would actually interact or not with this one. Now, the question became, can we actually automate this process? Can we actually find ways to discard lead molecules that can demonstrate biological activity? And more importantly, because these molecules are not that amenable to small molecule chemistry, we said, can we actually drive novel biologics? So biologics in this case happens to be antibodies, nanobodies, affitins, affibodies. You have a whole class, including molecular glues and things like that. I won't actually get into many of those details, but here I'll focus on one class. But that goes back to how we actually approach science itself. So you start with a set of hypotheses. You can sort of test that in the lab, and you want to assess that in the context of what you're trying to do. This has, simulations and experimentation that gets you new insights. Eventually, you report all of this in such a way that it is machine represented. And from those machine represented ways, you want to evaluate new questions and then eventually study that cycle again and again. But one of the biggest bottlenecks that we started finding was that this testing aspect of it is kind of very hard. And if you want to do this in the lab many, many times, it's actually a problem because these proteins have very different ways in which they work, and it's very tedious to do this. And as Francis was mentioning, directed evolution, as much as it is used, it is also a tedious process to kind of do it. It's very reproducible, but it's very tedious to do it. So we said, if there is history of lab automation, which is what you see here, tracked all the way from 1950s until the 2000s, where you can see very interesting work from Ross King's lab, where they did this with full-scale autonomy. You have a large history of what people have been able to do in terms of automation in the lab. But autonomy as such, which is very different than automation, is something very new right now. And there are a few companies and universities that have actually started doing that. You have agents for doing that in the chemistry side. You have Robin, which is actually from Future House, where they've done a lot of work with respect to building automated systems that can do the experimental data analysis and so on. In fact, they spun out an entire platform for doing that so that the scientific community can sort of use it for free. And there is another company called as Lila, which started to come out. There are other companies as well, like Periodic Labs and things like that, that are also doing the same thing, which is what they call a scientific superintelligence. In some sense, speeding up the way we actually do experimentation in the lab. But one of the fundamental limitations is how do we generate these protocols in the lab and so on. And this is the graduate work of my student, Rory Butler, who has been toiling at actually trying to think how we can run protocols inside the lab. And the idea behind that is we want to start with a high-level description of what the protocol is. Say a paper actually produces a lot of these biological protocols they're described. We want to extract that text. We want to be able to assemble and plan those steps, execute them, and see whether we can actually do that at some scale. So here is a full simulation of the exact lab that we are running at. So this is actual physical implementation. You can see the shaking because the camera is not that steady in the hand, but you can see that every step that is being produced here is being run or replicated. But this entire protocol was generated with AI or large language models. The code to run this was also generated by AI, right? And this actually starts up with this idea of we have a high-level description. You use AI and robotics in a way that you used to do for loop and plan motion, execute and run these. And it builds on a lot of approaches that have been talked about, that are going to be talked about here, right? So what we did to do this was build out an entire modular autonomous discovery for science platform. And that basically works with this idea that you could start with a very simple notion of your lab itself, which has a bunch of liquid handlers, robotic arms, and things like that. And they all talk to the individual devices, but you have managers that actually manage how the experiment is designed, how the events are being run, what work cells actually manipulate what samples and where they are, and so on. And this is all completely free and open source. You can use it as long as you have the hardware in the lab to do it. It's fairly simple to integrate within the workflows, and you can see a lot of people have actually contributed to this. And it's a fairly large model to actually like implement and work on, but it's made as simple as possible with different vendors as well. Now, in order to do this in a way that the agents could actually generate code and run, we took inspiration from one of the models that came out from Anima's group here, which is Voyager, which is based on Minecraft. I don't know how many of you played Minecraft. I used to play when I was younger. But Minecraft was actually kind of cool because it allowed us to basically say, you can have an agent with a prompt that says what the protocol should look like. You basically break that down into this hierarchy of tasks that it needs to do, and you can track the goals that are actually like doing. And in fact, this automated lab code generation agent is also like a Minecraft cartoon here. It was for a reason. But once you do that, you can actually simulate that using our simulation modules that I just showed you before. And then you have these programs that come out and tell you whether that execution ran fine or it had an error or not. If it were to verify the code, you actually store that in a skill set that you can recall at some point of time, and you allow the system to sort of evolve over the course of time to generate new protocols as well and so on. So this model that we actually built works in the context of having these things led by Priyanka and Brian, who are my graduate students, where you can actually see that it generates a code that basically tells you that it needs to move sample from one location to another. I'm just simplifying this. But as the agent is continuing to run, it actually discovers that it made a mistake. It tried to open and move the sample to a location where the plate was not open, and that it actually has to correct that. And it does that many, many times before it says, now I have the correct way to execute my protocol, and it does that end to end. So we are now in the process of finishing this paper up, which is called Prism, and it'll be available for anybody to use with the same set of equipments, with the same caveat again. If you have this sort of robotic uh, planning and tools available, now you can actually start doing even more, which is to how do we report this in a way that we can adapt these models in ways to understand new types of biology. So this is the context where we started building what we call as reasoning models. And this is actually an open source reasoning framework for structural molecular biology. Includes a lot of specific training data that allows you to do relational data distillation. So for example, when we are looking for new sequences and new types of data, we can actually go fetch them. It integrates biochemistry and biophysics into sort of tools so that you can actually like build on specific knowledge. And this is implemented entirely on high performance computing platforms using what is called Academy. And so in my world, where we deal with intrinsically disordered proteins or IDPs, we start with a target protein. Then we actually ask the AI to tell us what it knows about that protein by asking questions on existing literature. We use molecular simulations to kind of guide what that simulation knowledge would actually tell it to do. And then it also guides the experimental biochemical assays. It won't tell you exactly what the biochemical assay should look like, but it'll at least give you some knowledge on what background exists in terms of like validating those. And using that, we can now start generating new biologics. And I'll show you one example of that throughout my presentation. So this bioreasoner framework started with this whole idea that we needed very high resolution question answer sessions for biochemistry and biophysics. And we use this in the context of like generating questions from existing literature. So we use the entire ASM corpus, American Society of Microbiology, which is probably one of the largest collections of papers that are available on microbiology, and the PubMed open source papers, which is about 8 million papers. We had to parse that, we had to understand what was there. You filter for high uncertainty questions, and then for those high uncertainty questions, which is basically replicating how you would teach a graduate student or an undergrad student by increasing the complexity of the questions as it goes by, you mimic that process in such a way that it's actually like giving you more and more hard questions to answer. As you continue to do that along with tool calling, you actually get very good readouts. So here, as a preliminary result of like doing this training on the DeepSeek 8 billion parameter model, a specialized model for microbio, which is shown here in the green color, and the OSS 120B, which is the open source GPT reasoning model. This is comparing it with OpenAI. You can see that even with like lesser amounts of data, or lesser amounts of like training data, compared to the base model, this is actually achieving close to what we consider state of the art with these OSS reasoning models. So that is kind of cool, which is now letting us believe that we can actually train these models at scale. But once you have that, why is it useful for? So, for example, let's say I'm interested in this particular protein called WRN. I'm sorry, you cannot read the text here, but it's basically allowing the agent to infer disordered protein mediated protein-protein interaction. So say I don't know much about this particular target. I don't know anything about NMNAT2 or WRN or the proteins that I'm working with. I can actually ask this agent to fetch me all the data that basically tells me something about these proteins. In this case, I'm asking for a protein called WRN, and I'm asking it to tell me all the protein interactions that it is mediating. So it is saying that these are the proteins that it is mediating. There are several proteins that are listed. And in this particular case, it actually tells you that the acidic domain of WRN interacts with a protein called RPA and is phosphorylated by this other protein called CX2. If you take that example, it also tells you more information about the particular protein locations where these mutations might be affecting the protein's function, and you can sort of infer that automatically. If you get that sort of information, you now get constraints for modeling disordered protein interactions and their proteomes entirely. So for this case, if you take this protein called NMNAT2, this has a long disordered tail, which is about 75 residues or so. It's interacting with about 45 different proteins. It's hard for me to, connect all the 45 proteins and show it to you on one slide. But with respect to those 45 proteins, what you see here are there are distinct classes of interactions that are mediated. One happens to be this class of interaction where it forms a secondary structure which consists of a mixed alpha helix plus a beta strand, which is on this side. There is another class where it is purely beta strand interactions. And there are some classes where it is still completely disordered, but it's still very specific to that type of interaction. And if you use this sort of modeling, this one, so if you take AlphaFold 3 and tell you to model this protein called WHSC1, which is another target that we are working on, you can see most of this is actually like kind of disordered. Doesn't make any sense because the biological context was left out. And if you use the models to actually answer questions about their basic biological interactions, it now comes back with these structured regions that are highlighted. And from the quality of structure that you got previously, where it was completely disordered and you had a lot of low complexity regions, this now has very nice structure that tells you a lot about the interactions that are there. This big gap that you see here is the place where the DNA actually moves through the cavity and it's actually interacting with it. So I'm not showing the DNA for clarity. So if you have this sort of approach now, for the NMNAT2 sequences, I can start with a peptide sequence. I can now fold multiple conformations, simulate each conformation, cluster the conformations, diffuse peptide sequences that can actually go and bind to these regions, and analyze these energies for representation to kind of say, do I have a hit or not? Or I can use experimental techniques to actually like guide this entire process. Right? So there are various ways in which you can go about doing this strategy, but the Agentix basically allows us to explore multiple of these pathways simultaneously. And that's one of the reasons it's actually computationally very, very expensive to do this, which is that you have so many different pathways for analyzing the same protein in terms of how you would do the design for this. So if you were to take a look at this experimental data, we are working with Brian Dickinson's group, where they have this high throughput technique called the SPANCS. And if you're interested, you can read the paper there. But the basic idea behind it is very similar to directed evolution. So they try to evolve PPIs or protein-protein interactions using these phage libraries. And they have an initial library infection that's done in E. coli, and then they do conditional replication and so on, eventually leading to these de novo design of these binders. And for the NMNAT2 protein, they basically had like several binders that could have micromolar affinity. And in the case of disordered protein, micromolar affinity is specific enough that you would actually get reaction inside the cell. So that's actually kind of cool. You're able to do this at that level. But the interesting thing is you have data points now that are like 10 power 8 plus. Right? So you need ways to understand the mechanism through which these interactions are happening. And that's where these multimodal models actually play a very important role, where you have data that's now scattered in scientific literature, databases, experimental data, and simulations. You need to come up with ways to like combine that in ways that the model could actually tell you something about. So you might start with these preferred sequences. I'm calling CDRs because these are complementary determining regions. And using a method called as preference optimization, we can allow the language model to generate conditionally those sequences that we think would actually bind to those regions. In this case, it happens to be an antigen antibody, but it doesn't have to be really that. It could be any sort of sequence that you want to sort of fold. And you use partial diffusion as an approach to kind of constrain the generation of these sequences. Once you fold the sequences, you can use molecular dynamic simulations to gather metrics of whether these binding affinity actually exists, and is it something that we can verify with respect to the experiments. So one slight deviation from what I was planning to talk just about disordered proteins was these ideas of using how we represent DNA as such. So this is a central question that Frances also referred to in her talk about how do we encode things in the DNA. And one of the things that we decided to do was to build these foundation models, what we call as genome scale language models, again, with Animal's team and a whole bunch of people that went into doing this. But this is at the level of encoding the information at the gene using DNA. We could encode that using, codon level tokenization or other types of models. And now you can actually do this as a collection of genes or contigs. And that requires extending the context windows, which we actually demonstrated as possible using diffusion models, and eventually going towards full genomes where you can use byte pair encoding. But we cannot go to the level of full genomes like what EVO or EVO2 do. But at this scale, we can actually like understand the functional organization of the genome so that we can actually like tie them together. So this was the first version of the model, which came out in 22. And that model basically was a plain transformer architecture along with diffusion to kind of tell you how we could learn SARS-CoV-2 evolution patterns. So we scaled this up to like 25 billion parameters. It was still at that time one of the open models that are available. In fact, EVO and EVO2 actually use GenSlim as a way to benchmark their results, even though it's not part of the, it was not trained on any of those tasks really. But now we have come to the place where we can actually like include sequence pairs. Now we can actually do it at the level of the codon plus the protein sequence. That means that we now have a way to elevate this using a mass language model sort of an approach. It learns both protein and gene level representations, eventually leading you to some very nice insights about how we understand the entire gene organization itself. I don't know if this animation will play. Doesn't look like it's going to play. Hello. Okay, it didn't play. But what I wanted to show you there was that it could learn certain aspects of evolutionary details, including the fact that in spite of the fact we just gave the protein sequence or the gene sequence as the input, it could actually figure out isoform functions or orthologous function across different sorts of proteins. And that's where the starting point for the design with Francis and Anima's team was in terms of exploring TRIP-B designs. But this idea of using diffusion and DPO offers this explore versus exploit strategy, where the diffusion model actually increases sampling space compared to what we do just with the language model itself. And then the DPO allows us to focus on the sampling in a particular direction in the embedding space. So what happens with the LLM generated sequence, man, this is really hard. LLM generated sequences, all the gray spaces that you see here are just LLM generated. The diffusion generated model allows you to explore more of that strategy for like looking at a particular space. And the post DPO part of it actually allows you to infer even more along those lines of saying, okay, I want to basically focus on a specific region because I feel more confident about it from the experimental data. And that allows you to basically get resampling of the same set of amino acids, and their relative frequencies actually change over the context of whether you're doing pre-DPO or post-DPO. But what is more interesting is the fact that we can actually get 2 billion peptides simulated and tested and actually analyzed over the course of 10,000 nodes on Aurora. And this is not possible without optimizing the architecture for that, but we have been able to do that, and that's one of the reasons it's actually kind of cool. But for the same protein with NMNAT2, there are alternate binding sites that we wanted to target. So there are at least 2 binding sites, but you will see that this is going to get refined even more very quickly. So there are 2 conformations where we thought that we could actually like diffuse these peptide binders. And the AI models sort of reason from the interaction motives that it's not 2 binding sites that are complementary, but there are actually 3 sites that we need to be focusing on. So if you look at the models from the experimental data, there were only like 2 binding poses that were actually dominant, which I didn't show much, but if you just trust me on that for a second, there are these 2 binding sites that were primarily involved, B1 and B2, which is sort of interesting from the point of view of the binding sites that we were able to like target. But one of the evolution that ran and we got a sequence was looking at a complementary binding site across those 2 sites where the binding would take place, and is actually interacting with the same peptide motif. That's kind of cool because we never thought that that would actually happen, but it happened in the simulations. And we basically provided that input to the to Brian's group, who are now working at basically testing that out hypothesis and saying whether we have stronger binding if we include that binding site as well. And if you look at those motifs, they actually cover across the entire set of the interactome that was present, right? And that's kind of cool from the point of view of saying how these binding sites allow us to design these different target sites across the directions where we should be looking for new designs as well. So when you target that particular site, B1 and B2, you get an additional site that is for free, and you can use that to actually like now get even specific peptides that are now having high affinities as well. And you get very nice plots about the preferences for each of these binding sites with respect to their energies. These are quantitative measures that tell us about where we should be really focusing our attention on if you want to design these peptides. So these basic energy, basic versus energy or acidic versus energy tell us the distribution of the charges along those regions. And if you were to, focus on, at a particular region, you can generate better peptides, guiding the entire preference to actually align with what we are thinking in terms of the design strategy. And you can see with respect to the three sites and the distribution of the amino acids, what are the preferences for each of these amino acids in terms of how they, how they align themselves with respect to the binding site. So this allows us to diversify the libraries from what Brian was able to generate and give even more insights into how we should be targeting those specific regions. And this is done entirely by the reasoning model in some sense. It's basically simulating at each context and telling us how we should be thinking of these binding sites and how we should balance out the charge distributions across the binding sites as well. The last one I'm going to end up with is the prediction for the SARS-CoV-2 evolution, which is now taking this from an individual protein scale to sort of a viral genome scale. It's not quite at the level of an entire genome, but at least at the level of the viral genome, we can say a lot more. So this particular foundation model that we trained using bacterial genomes now allows us to fine-tune the model on SARS-CoV-2. And once you do that, you want to understand whether this allows you to basically like have new genomes that we can generate. And once you generate new genomes, you can now start doing standard measures of what we call semantic similarity and sequence log likelihoods, which basically tells you how likely are you to see these sequences in the wild. And semantic similarity score measures the similarity with respect to the embeddings that you get in that space. And these two actually correspond to fitness evaluation or immune escape parameters that now can be informed by MD simulations to kind of tell us whether a particular epitope would interact with the genome or whether it would interact with the spike protein or not in that particular order. And that allowed us to basically take a very small fraction of the year data. So we started with 2020 to 2021. So it has only seen 1 year's worth of this data. It has not seen the other year's data at all. So it doesn't know anything about the rest of the genomes as such. But taking this 1 year attempt and splitting the data, what we could actually find out was that the GenSlim fine-tuned on SARS-CoV-2 was able to distinguish variants across the entire plane. So I don't know if this video will play. Let's hope that it plays. Oh no. Is it running from a Windows by any chance? Sorry, you would have seen like really nice colors about the split about the genomes and where they come from. What is even more interesting is that these islands that you see here also correspond to something very important. It is the geographic spread of the virus. Again, these models only started with the genome sequences. We don't give it any additional input as to what the country is and whatnot, but it'll still tell you that there are some geographic differences, even though they are labeled as specific regions, specific variants and so on. So why is this sort of interesting? The Department of Energy has recently started reinvesting in AI plus biotech. There are several projects that are being funded across the DOE national labs. Anima's team and our team and everybody are part of it already. So there's like various efforts going on. The biggest effort probably is in this area where they are trying to do this as an orchestrated platform for automated labs with our group focused on protein engineering, applications, microbial phenotyping with PNNL in Berkeley, and plant phenotyping with ORNL. And there are various different applications. If you're interested in postdoc opportunities as well as opportunities in terms of working with the labs, please come and talk to me. But that's where this is all headed. Hopefully my summary has told you a lot. We are using these AI-guided reasoning models for biologics design. It can be used for enzyme discovery and design and optimization as well. Also for critical minerals research. I didn't talk anything about that, but that's actually a starting project. And now we are laying the foundation for autonomous labs, where we see the importance of having digital twins that are actually simulating actual protocols generated by the AI. And in terms of self-improving, which is actually a key thing for these models to actually be very successful, this is something that we are like working on. There are lots of math foundations needed to get that going in terms of like how we think about these models and what we can do there. And that's something that we are doing. Eventually, the scalability and hardware and software co-design, I think this is something that's kind of like working through the entire landscape of robotics providers, um, robotic-based companies like cloud laboratories that are already out there, and then eventually trying to see how we can actually train the workforce to actually achieve these with these new types of tools. So in some sense, I hope I've actually like convinced you that there is a lot of opportunities to use automation and robotics and AI in the context of these design problems, but that wouldn't be possible without the funding through the DOE programs, the CEPI, which actually funds a lot of the viral evolutionary landscape work, as well as ARPA-H for the biologics design as such, and computing time from Argonne, as well as other labs and all the colleagues and everyone. So thank you so much. Okay. So there are two ways to think about it. You can have the human intervene whenever. That's actually something that's given in the lab. We have seen some very interesting things happen when we just allow the agents to sort of evolve the whole thing if they want to do it. But we also have some restrictions in terms of how safety is handled inside the lab. So almost always, whenever we have the AI generate the code, it's inspected by the human first before it goes to actually running the lab. It would be a very bad thing if you just allow it to run by itself. We have seen some interesting events where we try to order materials from, of course, talking to the financial people and getting approvals and so on. We have tried ordering materials based on what the AI told us. I didn't talk about the antimicrobial peptide design side, where we were specifically asked not to do that, where the AI actually orders the materials on our behalf. That's a whole other story. But the safety angle allows us to prevent from doing that sort of aspect. But all the code that is generated by the models are first inspected, and then you run the simulations to make sure there is no issues in terms of tackling how the data goes back and forth, and then you run the code actually on the instrument. But it drastically narrows the time for getting one protocol functional to like chaining multiple protocols, which is very hard in the lab, even for humans to do. And now we are now in the process of like acquiring some humanoid robots to sort of manipulate, because we have a fairly large protein characterization facility. We have a fairly large crystallographic facility, and all these things have to come together. So, yeah. Thank you.